Okay, cool. Awesome. So I guess I'll start then. Um, okay. Hi, everyone. As you said, I'm Taylor Haynes. Um, I'm a PhD candidate at the University of Chicago in the Field Museum. And I'm studying parrots primarily, but particularly their chromosome evolution and their conservation. Um, the reason the title says Tales of a Wizard is because this process has been so interesting. Now I call myself a wizard because I actually survived. So working with avian genomes can be quite difficult. There's a bunch of features that make them difficult to assemble, and so trying to pick the best method protocols to do my dissertation was actually kind of challenging at first. Um, but GC content, repeat content, repeat content will come up at some point in this talk, and my least favorite type of chromosomes. Um, the first 10 to 11 chromosomes in any bird genome tend to be the macros, and then the rest are micro. And as you can see from this karyotype, which is which bird species specifically, um, they go from like medium to very small, but there's not much. You can see they get smaller, but you can't like it's not like gigantic differences. And microchromosomes are in a variety of different taxa, but not all taxa, as you can see from the figure at the bottom. So mammals don't have it, which is why I'm so jealous of people that work on mammals. Birds, it gets a little difficult. Now, a paper came out a few years ago on avian chromosome diversity, particularly in parrots, where this synteny plot shows how conserved the rearrangement in the genome structure is from the emu, which is the at the basal end of the bird tree, to the chicken, which is obviously one of the most studied organisms, and then going into passerines, which are highly studied and one of the most diverse group of birds. But then you cross into parrots, and it looks like a hot mess, basically. Um, there's a lot of things I call the flippity floppity, which is basically just things moving around, like going back and forth and just flipping places. Um, however, I want to note this only has four parrot taxa, and parrots, like there's about 400 species of parrots. About one third of them are endangered or threatened. Um, and their threats include loss of habitat, illegal wildlife trade, and hybridization, which is my favorite thing in the world. Um, and so this is a great system to actually study both chromosome evolution and try and actually use that data to help with conservation in some way, especially if what we learned about the chromosome structure and how that affects hybridization can inform conservation management. So just so you know, from like the placement on the tree, this is a whole UCE tree of all parrots. So cockatoos are at one end, macaws are at another, which I ended up falling into cockatoos randomly. Was always going to work on macaws. Now, if you actually, you can Google it, and there will be you can find an actual spreadsheet of the diploid number across bird taxa, which is basically my Bible. I just go to it, and when someone mentions a taxa, I look to see if something's in the karyotype, if it's there, and pull the diploid number. So when you actually pull the diploid number for the otta macaws, which are the most common, the ones that you think of, so it's the scarlet macaw, the green-winged macaw, the great green macaw, the blue-throated macaw, the blue and gold, Pirates of the Caribbean, if you don't know what. Um, and there is a somewhat of a variety of diploid numbers. Um, Typically, we think that the diploid number is around 70, but there are some sister taxa that have less chromosomes. So the scarlet macaw has 68, while its sister taxa has 70. The blue and gold has also 70, and the military macaw, which is the smaller of the green ones, actually has 66. Fun fact, they all hybridize in captivity with no seeable fitness decrease. And they can go for multiple generations too. I might have F7s in my data set, which still don't get. Now, using some nanopore data that we obtained and a VGP assembly, so the Ada Aruna genome is actually the one developed by the VGP. Ada glaucocalatus Ada rubigenes, and Ada ambiguus were all done using nanopore data. So as you can see from this plot, the macrochromosomes are relatively syntenic. There's a couple changes, obviously, but where the flippity floppity and the split and the fusions and the fissions happen is primarily in the microchromosome, which makes us believe that all the diploid number diversity is actually just in the microchromosomes, and very rarely you're going to find that in the macrochromosomes. Now, Kakatua, or Kakatuidae, the whole group of cockatoos, has a much wider range. And so the highlighted red tree, part of the tree is the genus Kakatua, which is the white cockatoos. And as you can see, it's pretty much within 70. There's 74, there's 76, there's 82. But there's taxa outside of that that are like 96. And actually, that one taxa, which is the gang cockatoo, can actually hybridize, and there are wild reports of hybridization with members of Kakatua. Why do we think this? Because cockatoos are almost interestingly boring. One inversion on the microchromosome. We have more species, by the way, of cockatoo. These are just the two that were done at the moment. With one inversion on chromosome 1, the diploid number variation seems to be just in the splitting of a chromosome. But there is still some rearrangements that might have some fertility, like some effect on behavior or any other form of reproductive isolation. So why do I bore you about the nanopore assemblies? Well, because number one, when we sampled them, they were all captive birds. So I literally, with the help of Dr. Connie Woodman and some others, went to a bunch of aviaries. So now I'm known as the vamp. Breeders go, when I say hi to people, they go like this, Taylor, cannot believe my birds today. But we went to a bunch of different aviaries and collected fresh blood, put them on liquid nitrogen, and tried a couple of other sampling protocols. However, with parrots, and this is the importance of museum collections, they hybridize in captivity a lot, especially when taxonomy changes. Let's say the citron-crested cockatoo has just been elevated, but it wasn't necessarily always a subspecies of the yellow-crested cockatoo, which they look incredibly similar. So sometimes breeders can accidentally pair birds up that are actually of different species, all depending on what your definition of species is. But also, not all species are available in captivity. Believe me, there are some that I would love to find and get fresh blood from, but I cannot find them no matter what I do. Also, a lot of permits. Almost all parrots are listed on CITES, Appendix 1 and Appendix 2. For those that don't know, CITES is the Convention on the International Trade of Endangered Species of Wild Fauna and Flora. So that's what outlaws elephant tusks and rhino horns. I always mix those up when I talking quickly. Also, parrots are incredibly difficult to catch. So the way around this was using museum specimens, particularly the tissue collections within Field Museum and other museums. However, when you're sampling very remote taxa, like on the South, the islands of the South Pacific, you may not collect the material, tissue, liver, lung, etc., and have it preserved in the best possible way that you would think of for long reads or linked reads. And so this is where it all comes to play. Or you also might not even get enough. You might get only a couple nanograms. So one of the major groups for my dissertation is the crimson-bellied conure, Pahura perletta, which was used from a really good, well-collected specimen in the Field Museum and was a female. And then its sister taxa, the pearly conure. Also, we had samples in the Field Museum, but they had not been accessioned yet. And we also used the female. Now, the biggest part of the TELL-Seq, we also have Hi-C data, and the Hi-C challenge was the extraction protocol and the steps. Because most times with museum specimens, some people can collect a lot of tissue, and sometimes people won't collect that much. And they might just collect like the tiniest amount, which for all the nanopore, PacBio, protocols, you need to say, oh, give us start with a lot of material, either tissue or blood, or then when you actually before you do the library prep, you at least need like 1 microgram of DNA. And so this is a tape station trace of the genomic trace of the high molecular weight of the Picora proleta extraction. And so it was about 50. We only ran this after the fact, after we did a bunch of runs, because, or we did at least one run and did a couple of assemblies because we noticed something a little bit weird. But we ran 5 libraries and Picora proleta worked. And then after running Supernova, did purge dupes, did 2 rounds of SCAF 10X, and then 2 rounds of Tigment, ARCs, and links to get the final assembly. And then we also ran it through Long Ranger to just double check on the fragment length and potential phasing for the stamp libraries that didn't work. And voila. The number of scaffolds was originally 64, it lowered down to 14, about 14,000. The longest sequence from the base assembly, or from the original Supernova assembly, was about 17 megabase pairs, and it ended up going up to 91.3. The N50 didn't change that much. The N50 was quite high, and as we'll talk a little bit later, there's a correlation between the fragment length and the N50 of it. However, the Pearly Conure didn't work, which is why I eventually got it to work, which is why I call myself the wizard. We also did the European Goldfinch, Carduelis carduelis, which is a project of Louise Boat in our lab. She's working on comparative genomics of introduced European goldfinches. That one was above 60, and apparently that one was collected in the same kind of conditions as the Picora proleta, where a dewar or a dry shipper was taken out to the field. It was put as quickly as possible after the sample had been killed. Now, we also did the American woodcock, Scolopax minor. And this is a project of one of our former postdocs, who is now a professor in Brazil, where he's actually working on the genomics and transcriptomics of migration and speciation. And this was actually collected from a fresh bird. So, like, we actually went out to a animal rescue, and they were actually having to put down birds, and so we actually collected it right after the bird was put down. That extraction didn't work necessarily as much, as you can clearly see. Um, and it was a little weird at first because we're going like, how did the museum specimens work and not the really fresh tissue one? Well, the extraction protocol that we were using is the homebrew extraction protocol that they actually use for the haplotagging paper. And I recommend to everyone, do it a bunch of times and always check before you start doing it on very valuable things. Because literally, the Picora lepida sample, the Pearly Conure, I went through so much tissue trying to get it to work. And so then end of last year, I decided, okay, I'm going to cut off a little bit because I've had very good luck getting enough to do a TELL-Seq library prep, and with high molecular weight, and it worked. Now, this is a couple of museum specimens, like the actual gel image from the tape station of a bunch of museum specimens that we did. So you can see that a lot of them don't show up. The actual Picora proleta is actually H1. There's 2 other tissue samples that are actually from other collections of different Picora species. Rarer ones, which I found very surprising, which actually really worked as well. The lepida is D1. So the Pearly Conure is D1, which wasn't that great of a run. And then the Carduelis was actually F2. So you can see that like this did not make me feel that well the first time. And the fact that 2 assemblies were really good, 1 assembly was okay, and then 2 we didn't get any data for. It kind of was like, okay, it's all about the fragment length. So Scolopax minor, Carduelis, and Picora proleta worked. And so when you map or when you put the like scaffold N50 to the contig N50 to look at the quality of the assembly. That red block is actually the Earth Biogenome Project's good metrics for like what is a good assembly. So it's a minimum of a 1 megabase pair contig N50 and 10 megabase pair scaffold N50. The nanopore assemblies that I presented a little earlier all fall into that quite easily. Quite easily. The TELL-Seq stuff, the TELL-Seq assemblies are almost there, at least for the two that were really good. So Carduelis is the one that's slightly lower, and Purpuretta is the one next to the green. And for the Carduelis, we haven't even done the post processing yet, so the N50 should go up at least a little bit to get over about 10 megabase pairs, I'm assuming. Now, the molecular length on the tape station to the molecular length that supernova outputs or long ranger outputs. They were pretty much near the same. And so that gave me hope for a good estimate for when we'll get some later data, which actually turns out to confirm this. And then when you actually look at the molecular length and to the scaffold N50, it correlates as well. So when the molecular length is low, the scaffold N50 is not going to be as good. So basically, I started making a cutoff because we have a larger project doing a bunch more. We did about 45 TELL-Seq libraries where I just ran everything on the tape station, and if it was below 60 KB, like 60 KB, we didn't use the sample. However, my extraction protocol works every time now, which is fantastic. That's when I tried Piper, the Pearly Conure again. So that Pearly Conure is F1, which, yes, it is a gigantic smear across the whole thing. However, there was a decent amount of high molecular weight above or even close to 50, 60 KB. The TELL-Seq protocol only requires, depending on the size of the genome, for a bird, it's about 1.2 gigabase pairs, only takes about 4 nanograms. And as you can see, the concentration when you actually do look at what's in the size range, it's only about 13.9 nanograms per microliter. And so we did a size selection using the Circulomics short read eliminator. And we got actually less DNA, but it was still enough out of the 30, 40 microliters that we eluded to actually proceed with a library. I have these ones up because these were samples that also gave me trouble, and the libraries was able to do high molecular weight out of them eventually, and actually good libraries. And so the one on the KU7143, that one actually was a very interesting story. The sample actually got sent to the wrong museum. And so during the evolution conference, our research scientist, one of our research scientists was walking through the lab at the University of New Mexico and then calls me going, Taylor, here's our sample on this table, not on ice. And I was about to travel for a trip, and I literally just threw my phone back on, I'm not dealing with this. After 2 attempts, the first attempt I made a mistake because my brain went on autopilot and I added the wrong elution buffer, which was very annoying. And then I threw it in the trash. Okay, my friend is clarifying that the sample was on ethanol, so that's why, probably. But it was still at room temperature, which is not ideal. And so I initially did the extraction, messed up the last step, threw it in the trash, went home going like this, okay, that's it. By the way, this is the only sample in the country for this taxa. Tissue sample, mind you. And so at that night, I was just sort of like, there was a little bit of tissue left because the digestion didn't just digest it all. Why not try again? And it's actually one of my better samples now. Point being, just because a sample may not be preserved at the best or most optimal conditions doesn't mean it won't work. Again, same with the other one on the other side. It's one of my Pahuras. It gave me trouble for a little while. Was collected the best they could. Because think about it, when you're going to the field and going to a remote area and you're collecting specimens, you're not always going to have time, especially to prep a bird or a mammal, to prep everything quickly and put them in minus 80 free or minus 80 or liquid nitrogen. So sometimes the tissue it gets left out over time versus immediately being collected and then put into a freezer. And so a variety of tissues that we've been using have actually been working fantastically. You just have to find the right extraction protocol. And then my favorite part was high-C scaffolding with the TELL-Seq assembly. Now, with the nanopore assemblies, obviously you have very few contigs or sequence or scaffolds at the end of the basis of the draft assembly. Um, and with this one, we had about 14,000. So this is when you map the high-C reads initially to the assembly. So it's not as messy as I thought it was going to be, but with um, one round of additional scaffolding and manual creation and some manual creation, it is a beautiful, beautiful assembly, with the exception of the W chromosome. The W chromosome is in about 11,000 pieces. And so the only thing that did not scaffold well in both the actual TELL-Seq assembly and the IC scaffolding is the W chromosome, which obviously we know W's and Y chromosomes have a lot of repeats and a lot of shared parts with the Z and the X chromosome. So that um, sorry, the fifth block down where it there's no blue around it, that's the Z chromosome. And so fantastically enough, I'm able to proceed with this. The only thing is the assembly is a bit gappy because it's short read based. And we're going to do some gap filling with nanopore, low coverage nanopore and or PacBio to fill in the gaps. Now, there's more projects that we're doing. Again, I said we're about doing about 45 samples, genomes. So kingfishers is a big one of them. And this is the one where I was talking about the tissues were incredibly difficult to work with until I got the extraction protocol right. And they were difficult to work with in the sense of I would run them once, but they wouldn't work. But there's not that much sample left. And do we really want to try over and over and over again and use up all the rest of the material that's actually in the collection? No. So that's why I say do an extraction protocol. The homebrew one from the haplotagging paper works beautifully and is dirt cheap. I love it. It is my favorite thing on the planet. Um, it does take a whole day for me. However, it is now that I've actually gotten the finesse of it, it works almost every single time on a variety of samples. And the Pearly, which actually I didn't quite explain, that was collected on liquid nitrogen, put into a minus 80. The minus 80 died, got moved to a minus 20, and then got left in the minus 20 for about 8 years. And so that's why you saw that all that shearing. But I wasn't able to get any of those larger fragments the first time around. It was all shorter fragments. So both of these lovely people are going to be using the Tody Ramphus Kingfisher data that we're going to be generating. And so I'm going to close with just pricing. So a nanopore flow cell costs about 17 to $1,800. I was going to say 1,000. My brain always adds zero for some strange reason. Now, because this is an Illumina-based technology, and a bird genome is about 1.2 gigabase pairs, you will need, and you cannot fudge this, about 55 to 60x coverage, because supernova or anything else will not. I've already even tried to run some of the parrots with TELL-Link, the lower coverage ones, and you do not get a good assembly whatsoever. However, duplication rates. So our duplication rates in the first round were incredibly high, like over 50%. After talking with the lovely people at Universal Sequencing, we were able to just figure out that we just have to split the library pool where you would PCR amplify, and then put it back, split into two, put it back together. And now our duplication rate is just on the high end of what they would expect, which is about 35%. And so accounting for that loss in duplication, you can fit about 4 genomes on a NovaSeq X 10B lane. And the new NovaSeq X1B flow cell, which I can't wait to come out because it's just so much cheaper and smaller, you can fit about 4 genomes, which is 2 per lane. Considering the kit is the standard kit, which for large genomes, anything 1 gigabase pair or higher, there's 4 reactions, so it's about $225 per sample, if you think about it that way. And then when you actually divide what the pricing of the lanes are, which at U Chicago, the lanes are about 1,400-ish, you get about $500 per large genome at 55 to 60x unique reads. You will have extra, and doing that math has actually worked perfectly for us. So when we're doing some low coverage stuff to do phasing, to look at recombination rates in these weird hybrids with different chromosome numbers, and doing assemblies on any sample really that comes out with especially low yield, when it comes to the high molecular weight protocol. I haven't had a lot of them, but there are very few. There are some, and they're rare taxa that are coming up to be like, oh, it will only get 2 nanograms per microliter, and then I just dry it down and to run the tape station. There's high fragments, there's large fragments, and then I just add 4 nanograms to the library prep. I spent a year optimizing the library prep because, as I've said to some people, it's a kind of find your own adventure protocol, which was kind of scary at the very beginning, but it's not as scary as you think. And I can do multiple samples, library preps from start to finish in 4 hours, which makes my life so much easier. Instead of multiple days, like my IC, using the Dovetail OmniC or the Arima kit, it takes multiple days. So actually, next week I will be doing a couple TELL-Seq libraries and then dropping them off the same day. I mean, they really work, which I've gotten about a 95% success rate when it comes to getting libraries to work. At 5%, I literally accidentally forgot to add one of the enzymes. So thank you so much for listening. And does anyone have any questions?
Yeah, anyone who have questions, please unmute yourself and then you can start ask questions.
Hi, Taylor. Thanks for the presentation. I had a quick question around data analysis pipelines you used. I know that TELL-Seq has some off-the-shelf options. I might have missed it before, but were those used in any of the data analysis that you use from the files coming off the NovaSeq, or did you also develop any of your own pipelines? I might have missed that part a bit there, so I apologize.
No, sorry, no, that part. No, so I used universal sequencing's pipeline to basically, once the stuff comes off the sequencer, then you run it through TELL-Read, which is actually really easy to run and really fast. And then it will put it out in a format that you'd have to, a cleaned, filtered format that then you then convert to a 10x format. When I first started putting together my pipelines, TELL-Link apparently was not built for larger genomes like mine, so that's why I went the 10x route. But obviously, also most of the pipelines that other people have used that I just kind of pick up are 10x. I told a couple of my lab mates that we're generating the data for that it would take a while. I actually just did it recently. I was able to get from coming off the sequencer to converted 10x format in a day. I'm hoping that answers your question.
Yep, that is perfect. Thanks a ton, Taylor. I guess then also as a follow-up, do you have any future plans for using TELL-Seq with any projects that you're currently planning? Um, either around, any other kind of conservation purposes.
It's so the one big concern for working on the hybrids is that in captivity, like let's say subspecies, when you get to subspecies level, like for the military macaque, there's three subspecies. There's the Mexican, there's the normal, and then there's the Bolivian. I can't tell the difference between any of them visually. Most breeders can't. Um, and so considering there's so much diploid variation in between species, I was going like maybe there's some diploid number between subspecies. Um, and so I was actually able to find from LSU a bunch of tissue samples across the distribution that actually worked really well. So that we're going to be doing TELL-Seq so we can get phased haplotypes. So we can actually do some local ancestry work um, and actually have a representative haplotype that's like phased from start to finish across a couple birds. So that like I'm so excited that I'm actually going to have a full haplotype, so I actually know where the haplotype looks in one of these species.
That's great. Thanks, Taylor.
Yeah, for those who cannot unmute yourself, or you can use the chat to type in your questions so we can also see it and answer it. So Taylor, thank you very much for such a good presentation. And I have a question regarding to the DNA extraction paper that you referred to. Is there some particular paper that the protocol that you use that it's so good on extracting the high molecular weight DNA? Can you refer it to us by either typing in the chat or show it to us on the screen if that's not too.
Actually, that I will show. I will show. Let's see if I can show it to you. Because it is.
I bet people who are, trying to, take advantage of the TELL-Seq would love to see the way that you is successfully using to extract high molecular weight DNA, because it looks like it is a very important step to make
Oh no,
sure
that
that
was the
the success.
no, that was the biggest hurdle. And so again, so what my recommendation for anyone that's doing a bunch of museum specimens is can get used to the protocol extraction protocol first. Don't just, especially if you have grad students, don't just go, here's the protocol, do it. Because I got better over time. And I guess it's just those weird things. It's like when you're cooking, you know how fast to do something, or it's I literally say people, lab work's almost like cooking. You just can't taste what you're making without because there's no taste in it, or you might die. One of the two. Um, let me. This is the paper
Oh, I see.
that I took their protocol from. And so it's a bead-based protocol. So if anyone uses speed beads, um, the recipe is a little different from when you make regular speed beads for when you're doing like a whole genome prep or a rad prep or something like that. It's a little bit different, so pay attention to it. Don't just think, oh, regular speed beads. So I literally have a tube of high molecular weight extraction beads. Um, Also, I don't know if this is just bird material because birds are just a little annoying when it comes to sticky proteins. Um, when you finish the extraction, like when you're doing the bead washes, do it slowly, because otherwise you'll take up all the beads with most of your DNA. But the DNA will probably still have some of that brown buffer in it until you dilute it or wash it even more times. So if it looks brown, it's still good. Um, and I only use this protocol on the tissues because bird blood has a lot more of those proteins, and so the beads just take up everything. The nanobind protocol, like when you're actually had to do them both at the same time, like on two different samples, the reagents are almost all the same. The exact almost all the steps are the same, And so the only difference is one uses a magnetic disk, which really helps when you have those like those sticky samples, and then one has beads, which therefore, like I was losing a lot of the samples when I'd be like, oh, pipette, oh crap, everything's in the like in the stuff that you're discarding. And then I would thought that would actually like kill it, but it once I started learning, like, okay, do it slowly, use smaller pipettes so that you're not taking up all the beads. Then I started getting, and also never only run on tape station, or if you have a pulse generator, do that. Don't ever try running on a gel. Yes, even if you use a high molecular weight ladder, do not run it on a agarose gel. That can give you false readings. That's what we did the first time, and just like a lot of the samples were actually when we did the tape station, they were very different.
Great. Thank you so much, Taylor.
You're very welcome.
So is there any more questions? All right. So it looks like we are very effective and efficient on this call. And then I would love to
Yeah.
give some precious time back to everybody's life. And thank you, Taylor. For those who are interested in getting more TELL-Seq related questions, please feel free to send us an email or give us a feedback on the chat. So I'm going to stop recording here.